Cell division. We will discuss about the cell division. Cell division are of three types: amitosis, mitosis, and meiosis. When the injured in the case of injured cell, the cell is divided in unequal part to unequal daughter cell. This is called amitosis. Mitosis are the general cell division. It is also called the equatorial division, where a cell can divide to form two identical daughter cells. And in meiosis, a cell is divided to form four haploid daughter cells. Here we discuss about the mitosis. What is mitosis? Mitosis. Before the cell division, there is the question: why cell is divided? What is the reason? First, we discuss about the reason why cell is divided. The reason is that when a cell is divided to form daughter cell, in daughter cell, growth, development are take place, and finally, in the amount of nucleus or the number or the amount of the DNA, it become double by the process of DNA replication. Finally, after that, in cell, the amount of the DNA become double, but the number of chromosome become same, and ultimately the cell is stable until the ratio between the cytoplasm and nucleus can be constant. A constant ratio are found in stable cell when the nucleus and cytoplasm is found in a fixed ratio. When the amount of nucleus is double, ultimately the cell is unstable in a reference to the nucleus. So first, once again, the unstable cell can regain the stability to divide their nucleus, and ultimately in a cell two nucleus are found, and this process, the division of nucleus, is called karyokinesis. And after that, here once again the ratio of the nucleus and cytoplasm become disturbed. Once again the cytoplasm will divide, and finally two daughter cell are formed, and this is called cytokinesis. So we can say in mitosis there are two type of the division. One is the karyokinesis, another is the cytokinesis. First is the karyokinesis and second one is the cytokinesis. The whole process of karyokinesis are further completed into four phases. That is, first is the prophase, second is the metaphase, third is the anaphase, and fourth is the telophase. For the easy understanding and learning, pro means preparatory or the first phase, meta means P phase, M phase, and T phase. This is the terminal or telophase or tailor part, middle part, and this is the primary or first part. Here we discuss the process of karyokinesis or the four step of the prophase. How can it occurs? First is the prophase. When prophase started, at that time the cell nucleus is interphase nucleus. When there is no division, there is no any change, and then the nucleus of interface is look like this, and the structure is, this is the cytoplasm, chromatin thread, nucleolus, and centrioles or centromere. In prophase, the interface nucleus become change, and the first change is occur. The chromatin thread, that is look like a thread, it become a visible structure and possess the arm, and these arm are joined with the centromere or kinetochore, and finally if specific shape structure is converted, and this is carried out by the process of condensation. So first step, the first change in prophase is the condensation of chromatin thread are take place, and finally it can be converted into a visible shape structure. Second, the nuclear membrane. This is the nuclear membrane. This is disintegrated in the cytoplasm or disappear in the cytoplasm. Third event is the nucleolus. It is also disintegrated in the nucleoplasm, and finally the centriole or the centromere, which are duplicated at S phase, they can move to the opposite pole, and finally the ester race formation or take place, or spindle formation or take place. Second is the metaphase. In metaphase, certain change occurs. First, when the cell enter in metaphase, then the nuclear membrane is totally disappear. Centromere reach to the opposite pole. Here, two type of the spindle fibers are formation are take place. First, that is the continuous, and second, all the chromosomes at that stage they can possess, each chromosome are bivalent, and this is the kinetochore, this is the kinetochore. They are arranged in between the two pole, or in between the mid pole, mid face plate formation are take place. Finally, like here and here, this is, sorry, they can yeah, एक और होना चाहिए चार, once here means at the metaphase all the chromosome are placed in between the two pole. In reference to spindle fiber formation, there are two type. This is the chromosomal spindle fiber, and this is the non-chromosomal or the continuous spindle fiber. The continuous spindle fiber are attached from one pole to another pole, and finally at the metaphase the chromatin thread can be visible to a very unique structure that can possess two arm tetravalent and This structure was called the chromosome by the Waldeyer. Next is the anaphase. In anaphase, the main event is the segregation of the chromosome. These chromosomes are segregated. You know that the chromosome, this is the chromosome, and the chromosome are attached to the pole by the centromere near the kinetochore. So the separation of these chromosome are take place, or we can say that the division of the centromere are take place. And finally in anaphase, finally in anaphase, this is the pole. Here the stretching in between the continuous fiber are take place, and finally slowly these chromosome with two chromatid are moved to the opposite pole, and finally at the end of anaphase each pole can possess half number of the chromosome in reference to chromatin material. In number of the chromosome, each pole can possess equal number of the chromosome. And last event is the telophase. In telophase, all the events that are occurs in prophase are occurs, but in reverse manner. You know that in telophase, the first visible change is occurs that in a telophase, this is the telophase, the nuclear membrane is appear, both the chromatin. Finally, nucleolus is also appear. The chromatin, the chromosome can then recondense and convert in a thread-like structure, and at the end of telophase, a cell can possess two independent nuclei, and finally the process of nuclear division are take place. After nuclear division, the division of cytoplasm are take place, and they are differ in plant as well as in animal. In the case of the plant, animal, in the case of animal, cytokinesis are take place by formation. In this case, the cell that can possess two nuclei, here the invagination or furrow formation are take place both side. It can be deepened from both end, and finally they can lead to each other, and like this finally, and after that the cytoplasm is divided, and a cell can be divided to form two daughter cell. Whereas in the case of plant cell, the cytokinesis is differ, because in the case of plant cell, the outermost boundary is the cell wall. So in the case of plant cell, plate formation are take place. Here, the deposition of primary cell are take place in between the cell, and finally, when the cell wall is formed, then it can be segregate or separate, and finally, two daughter cell are formed. So, at the end of this, two daughter cell are formed. So, finally, we can say in mitosis cell division, a parent can be divided to form two identical daughter cell, because in parent as well as in their daughter, the number of chromosome is always same. They are identical. So all the growth and development are take place by the special cell division, that is called the mitosis. Thank you.